This is kind of a long title, but the idea is that we are working with this. I've been working with this some of the materials from caseitproject.org, and there's the website for that, and that's a system that was developed by Mark. And so I'm going to go through relatively quickly an example of something that he's developed on there that I've used in a class. And then at the end, he can give a broader look at some of the other things that are on this, that are involved with this project. But overall, they're case-based experiments using a variety of techniques. And lately, there have been a few that have been developed that go through primary papers. And Mark selected really, major papers to go through. And so I'll go through the one that I used in a class relatively quickly, but then we'll have more time later to talk about the details. So I did this with a class. It was a kind of an ideal class to do something like this. It was a seminar class. It was, the title of the course was Genomic Engineering, so it was basically CRISPR-based class and sort of a journal club style class. And so we did a lot of CRISPR papers throughout the class. And the first one we did was the one by Jinnick et al. That led to the Nobel Prize for CRISPR. And the idea is to repeat the experiments using the software that Mark's developed for the students to sort of dig in to the experiments and try to think about how they were done and sort of replicate the experiments. So each student was assigned a figure. I don't know how to get rid of all that stuff, so I'll just have to say there. So usually when I do journal clubs, I just give students a figure so that they can, break it down into bite-sized pieces. And they all recreated the figures, and then they presented the background for that figure and the original figure, and then compared them and made a presentation to the class. And I broke it down. There's the assignments, the paper is really broken down on the website through CaseIt into discrete steps that were easy to break down for students. And so this is the paper. And it's called Programmable Dual RNA Guided DNA Endonuclease, which is, I like this part of this that I like about this paper, other than it's, one of the most important papers ever, is just the title is really descriptive of what they had. So the idea that it's programmable, that there's 2 RNAs involved, it's a DNA endonuclease. I like descriptive titles like that. And so just a little background on CRISPR. Maybe we can move that out of the way. I'm sure most people know this stuff already, but the components of CRISPR are important for these experiments. So CRISPR is this sort of locus in bacteria that expresses one big RNA that has these spacer regions in it. And then these get processed into a CRISPR RNA. And it's these spacers that target DNA to be cut. And then there's this other DNA called either tracer RNA or tracker RNA, whatever they call it. This also has to be expressed and packaged with the CRISPR RNA, along with Cas9 endonuclease to cut target DNA. And it's these spacers that determine what the targets are. And so the things that in this experiment were manipulating these spacers was one of the things they did. So this is what it looks like when the RNAs match up with the DNA. You have the CRISPR RNA and then this tracer RNA, and they sort of pair up. Then there's this other sequence called PAM, which is important for defining a CRISPR target site. So all of these things were discussed in the paper. These are all the major findings of the paper. We went through all of these in the class, but I'll just do these 2 in this talk. And so just the basic in vitro system where they were able to get CRISPR to actually cut DNA in a specific manner. And then how they were able to put those 2 RNAs together and make a chimeric RNA. And then they applied this to different targets within this plasmid that contained the GFP gene. That could have been anything, but that was the plasmid that they used as a target. And that showed that this was programmable, which is a really important part of the system. And so I broke it down into these different figures. And again, we'll just talk about 2 of these. And so the first figure, they had this different plasmid, not the GFP plasmid. And these plasmids contained targets for the different spacers. They call them protospacers. And there's protospacer 1, 2, and 4, but this one used 1 and 2. And so basically the gels, they run the plasmid uncut, and then they cut it with a spacer that would, a CRISPR that contains a spacer that won't cut it and one that will cut it. And then they linearized the plasmid and repeated the experiments with the linearized plasmid. So this is what the paper looks like in, or the figure looks like in the paper. And the plasmids were all supercoiled. So when they're uncut, you get 2 bands. And then when it gets cut, you get 1 band. And so this is the key experiment here, where when they put all these things together, when they put spacer, endonuclease, the CRISPR RNA, well, that's the plasmid, the CRISPR RNA, the tracer RNA, and magnesium, it cut and you get a single band. And then when they linearized it first and then cut it, then you get your, when it's not cut by CRISPR, you get these bands, single bands, and when it's cut, you get these 2 bands. And so that was one of the figures that they were supposed to recreate. And so these are the things that we're going to be looking for. And this is what case it looks like. And so you sort of pick, so your DNA, they come in just forms of files that you pick from a folder, and you have your, there would be further up in this window, there'd be just the DNA. And then then you cut the DNA with these enzymes. This is the XHO1 to linearize it, et cetera. So you do all your manipulations of the DNA and it shows up in this window. And then you load these into a gel over here. And the things that are loaded show up in this window. And then you run the gel and you can stain it. And so the gel sort of looks like this. And then they cut out their gel and put it into a PowerPoint. I couldn't find my students' actual PowerPoint presentations. That was the idea, to use theirs, but I just recreated one myself. And so this is all these things. This is uncut plasmid. This is plasmid cut with non-specific CRISPR enzyme, specific CRISPR enzyme, then the linear plasmid, the linear with the non-specific CRISPR, and then the linear with the appropriate. And so then so this just shows that they can cut it, but what the students did, so they had to go through this and think about it and recreate this image, and then they compared it. So then they took that image that I showed before, where we've got this one that's cut and this one that's cut, and then they had this, I made them go through this, where they say this band, and these bands correlate. So they had to recreate those exact bands and explain how they were derived. The second experiment is where they used a chimeric RNA. They put 2 RNAs together, and then they targeted different regions of the GFP gene. And so this is just to show what they mean by chimeric. So here up in this top figure, there's the 2 RNAs that they need, and they literally just linked them together. They just expressed them as one RNA and showed that worked. And so now that's what everybody uses as a guide RNA. And so then they made these RNAs to target different regions of GFP. So GFP is in this plasmid, and then there's a SAIL1 site here. So they target it and they cut it somewhere inside GFP. Then they cut it again with SAIL1, and then they generate these little fragments. And so this is just showing what those targets look like and what the RNAs and the CRISPR RNAs targeting them are. And then this is the figure that was in the paper that showed that these different targets got cut and gave different sizes. And so so they then had to replicate that and show that they just had to redo the experiment using the CASEIT software and come up with a gel. And you can see that this pattern is the same, with these 3 bands sort of increasing like that, and these 2 here. And so this showed that that CRISPR is programmable, which is the key thing about CRISPR, is that you can target anything for genome editing. And so the students really sort of got into this. That whenever you're doing stuff with computers, there's always little technical issues of how to get it to actually work, and they expect everything to work immediately. But once they got past that, I think they like the idea of going through it like this. And what I got out of it from the students' perspective is that it forced them to focus more on the details and forced them to understand the experiments, and they really seemed to have a good understanding. And then we went on and did additional papers throughout the semester, and they really did. They really knew what they were talking about. They knew what a PAM sequence was. They knew exactly how CRISPR works. So when we looked at other papers, it worked well. And then there are some other primary papers that have been already developed. One is using CRISPR to treat an actual disease in humans. And the other one is the paper that led to these mRNA vaccines. I worked through the top one, but not the bottom one yet. But these are also really super important papers that give students an opportunity to really dig into those. So I just wanted to give you an overview of that and make sure it was quick enough. So I gave Mark a few minutes to talk about the overall site. So I'm just going to stop there and stop sharing and let Mark take over.
Okay. Hi, everybody. Can everybody see my screen?
Yes.
Okay. Well, thanks very much. Ken gave a nice overview, and I just got a couple of minutes here. But the CASID project is a very long-running National Science Foundation project. It's been going for actually quite a few years. And we have a whole series of cases based on primarily on genetic and infectious disease. So you can select sickle cell, for example, Huntington's disease. And I have mobile versions, for example, sickle cell, where you can link to the nature of the mutation and you use NCBI tools like the Genome Data Viewer and Structure, which is another online tool that you can use. But for the purpose of this talk, I just wanted to mention the papers that Ken was referring to. And this paper actually is one of the most, in my view, one of the most important papers in biological history, because it did win the Nobel Prize. And there's versions of CASID that you download for both PC and Mac. It's a PC application that you can run on the Mac. But because it can be a little cumbersome to use that when you have limited time, I do have mobile versions, especially of the genetics cases. And I'm planning on converting everything into mobile versions. And by mobile versions, I mean basically screencasts of the software running. So for example, here is what Ken was talking about. This is about a minute and a half. This is actually part 4. This is how you reproduce part 4. So this shows, for example, selecting the non-complementary strands and then cutting them, and then selecting a control, and then cutting that with the complementary strands with a Cas9 enzyme. And I'm just to speed, since we're limited in time here, I've only got about a minute. I'm just going to scroll through this quickly. But this is what the students would do with the actual software. And so I'm just going through all those steps very quickly, and then you stain it at the end. And again, that's one of the most important figures in history, and it won the Nobel Prize. That particular figure, which is figure S15D in the supplement to Genix paper, is really what won the Nobel Prize. So if I go back, this mobile version is a highly condensed version. This is what Ken was talking about, but very quickly, in these exercises, you have a series of questions that are designed for students to think. There's the messenger RNA exercise, again, that he mentioned, that basically is the reason we can all have this Zoom meeting because of the COVID vaccine. There's the action potential experiments, which won the Nobel Prize in electrophysiology for Hodgkin and Huxley. Very important stuff. And at the end here, students can actually watch.
Martin Jinek in the lab made that construct, and he did an experiment, a very simple experiment, to test whether we truly had a programmable DNA cleaving enzyme. And the idea was to generate short single guide RNAs that recognize different.
So again, that just shows the significance of this. And in my view, it's a really powerful way of doing it. And very quickly, I know I'm over time here, but I'm just going to show a little bit of the evaluation that was done. Now, I'm no longer teaching, I'm retired, but Ken did this, and one of my colleagues at the University of Wisconsin River Falls did it. And so here was a feedback exercise that we gave them, the students. And then here are some of the responses. And for example, how well prepared were you to use the exercises? Which of the following topics should be explained in greater detail? So these are the responses, a summary of the responses. How would you rate the value of the CRISPR exercise as an educational tool? What do you like and dislike about it? And then the ATTR exercise I didn't talk about, that was the first use of CRISPR. What did you dislike or dislike? I love the idea of open-ended research that was talked about in the general suggestions. Clicking through the other cases, I thought, blah, blah, blah. many places where they could be used. I think coming out of the time of online classes and the challenges that come with it would have been a super awesome resource to use among a lot of my courses. Of course, I'm picking out one of the better comments. But I realize I'm out of time here. I went a little bit over, and I do want to allow a few minutes for questions for Ken and myself.
Yeah, so we have a quick one. Can students manipulate the factors to get different results, to ask what if questions?
CASEIT is totally open-ended software. It'll work with any DNA or protein sequence. There are lots of techniques besides restriction enzyme digestion, southern blotting. There's microarrays, for example. But for those particular papers, I was interested in just in coming up with the original sequences. Those are actually the original sequences that Jennifer Dowden and her colleagues used. I got them from AdGene. Those plasmid sequences are genuine sequences. So it took me a long time to develop that, but that was specifically to recreate those experiments. But you could use CASEIT to do anything because it's totally open-ended.
Stuart, you have a question?
Yeah, I was just going to say that I teach a recombinant DNA technology course, and we have a lab component where we actually do CRISPR-Cas in it, and we utilize tools like DNA Strider, serial cloner, things like that. And so I was just wondering if your software is a lot like those types of software.
Well, CASEIT has a specific purpose, and the techniques, you can visit the webpage and you can see the techniques that it used. It's not really designed, it's more designed as a teaching tool. It'll work with any DNA or protein sequence, but I'm not trying to keep up with all of the latest advances in technology.
Sure.
So these are some fairly historical techniques that have been used, but what I'm doing with the mobile versions is I'm for example, sickle cell or Duchenne's muscular dystrophy. What I'm doing is linking to the NCBI tools where students can really get into those like structures, see the three-dimensional structure of the hemoglobin for sickle cell, for example. So the goal is to use CASEIT as kind of a shell, kind of an introduction to branch out into some of these other tools. So you start with some interesting historical Nobel Prize winning case, let's say, or one of the other cases, and then you branch out from that. But it's not designed as a research tool for current techniques.
Mm-hmm. And I was just scanning through the CASEIT website while you were talking a little bit. And so the idea behind each one of these is to actually go to a specific paper and recreate some of the data within.
No.
Is that correct?
The only ones that deal with specific papers is the one that we just talked about, the ATTR, and then the 2005 paper of Crico and Weissman. The other cases are cases that we developed. We did role playing with students. These are like hypothetical families. And so the students would role play as family members and as research technicians and as genetic counselors. And we had a whole system, a discussion board system where they could create posters and then discuss the posters and then switch roles. They would actually, when somebody asked questions of their poster, they played the role of a family member, or rather of a research technician or a genetics counselor rather. And then when they asked questions of another poster, they played the role of family member. So that was really separate from the original literature. I just in the last 4 years or so, I got involved in adapting to this to class paper. I mean, really important papers, either Nobel Prize winning papers or papers that should have won the Nobel Prize, like the 2005 paper that didn't win it and it should have won it, as a way of really engaging students.
Okay, and I'll just ask one super quick thing and then shut up. But I was just looking at like your colon cancer example here on your website. Do you provide them with some of the data with respect to like the protein sample for Sam's tumor tissue and normal colon tissue? Because I don't see like files or anything for them on the website.
You have to, to use this, you have to download the CASEIT package, either for the Mac or PC.
Okay.
And then the files are all part of the package. So they
got it.
They use these are real sequences. We're not made up.
Okay.
The protein's a little artificial, but the DNA is all 100% real.
Okay, but it's in the software package. it's in the software package that you would download.
Yes, yeah, you're right. It's
Okay.
a simulation
Perfect.
that, right, yeah.
Yeah. And it's,
Okay, thank you.
It's a very simple download. You just download the package and unzip it, and then you're ready to roll. And I use the cancer one, the microarray. So another way to use it is, so you run a microarray and you have breast cancer, colon cancer versus normal tissue, and then it's a small version of a microarray, and you get some genes are overexpressed, some genes are underexpressed. And I just have students sort of, if you click on one of those genes, you can actually get, or just one of the spots on the microarray, you can get the DNA sequence, then you can have them do a blast. You can actually have them do that straight from the CASEIT software if you want, and then have them work that up as, what is this gene? Why would it be involved in cancer? That kind of thing. So you can, it's sort of like simulating, it's a way to sort of like simulate an experiment to get some data to then sort of talk about it and analyze it. And you can take it wherever you want from there. And,
okay.
And for the person who asked, so I think it was Carol that asked if you could change it. I think you can. I haven't really made my own cases, but you can change the files that you use for probes, or that kind of a thing.
It's totally open-ended.
Yep.